In our latest study, we combined synthetic biology and cloud computing, two incredibly powerful techniques to create a new chikungunya vaccine candidate. Our approach is not confined to chikungunya alone, but can cover the entire universe of infectious diseases where antigenic epitopes exist.
Half a century ago, many people had started to think that infectious diseases had been solved, and we were totally wrong. Infectious diseases have never been more of a problem than they are today.
Our best defense and most successful defense against infectious diseases is vaccination.
Chikungunya is one of a group of viral infections that's spread around by mosquitoes.
It is usually confined to sub-Saharan Africa, but because of deforestation and climate change, it has started to spread all over the world, also reaching Europe and the US and all developed countries.
And it's just one of a whole bunch of these infections against which we have no treatments and no vaccines at present.
A major problem with vaccines at the moment is that they need to be refrigerated for storage and for transport. Otherwise, they become inactivated. We have developed a new class of synthetic vaccines. We call it the ADDomer. It consists of a protein which we have engineered so that it forms a large particle structure resembling a virus.
This technology is unique because it doesn't rely on a cold chain. We can mass produce it at low cost, which is basically ticking all the boxes in what a very good vaccine should be.
This is the ADDomer. We have applied synthetic biology to engineer the surface of the ADDomer. By putting small and harmless bits and pieces of the chikungunya virus on top of the surface of the ADDomer, we create a particle which looks like chikungunya but is not. But when the immune system sees it, it develops antibodies against it, which will be protecting also when the real virus arrives. In order to be able to do so, we had to know the structure of the ADDomer at near atomic resolution. This we determined by cryo-electron microscopy, and we applied for the first time cloud computing to compute the structure from the data which we got out of the microscope. And we got a very strong immune response in our studies, which showed us that we are on the right way.
I think this is an example of really a step change where up until now we've had to kind of accept the materials that biology gives us. This represents a more deliberate attempt to actually engineer the bits and pieces that you need to make a vaccine work.
This was a highly interdisciplinary study involving many scientists with complementary expertise. In Bristol, we have a unique ecosystem for this kind of work, which is hard to find as well. The IT team here in Bristol, together with their colleagues at Oracle, did an excellent job to connect the data which comes out of the cryo-electron microscope with the cloud infrastructure.
The cloud computing fundamentally is the ability to be able to get computing or storage or networking access as a utility. This was a very interesting project. The amount of data that was required to be processed and the complexity of the computational models required a fairly novel approach to be able to use different kinds of hardware and cloud infrastructure for different parts of a complex workflow. So what the team here have done is demonstrated an extremely cost-effective and fast way to analyze cryo-EM data. And even better, the research team at Bristol have published their work into open source, so it makes it very easy for other groups working with cryo-EM to be able to leverage that.
So what cloud computing does is allow us to dynamically create resources to fit the exact problems that we're trying to solve. So the tool we created is called Cluster in the Cloud because it allows anyone to create their own personal high-performance computing cluster in the cloud.
What we need to do now for the next steps is to continue the validation in other infectious disease area and to continue to develop our technology.
Our ambition is to make vaccine candidates based on our technology against a whole range of human and also veterinary diseases. In our current paper, we already show more than a dozen other vaccine candidates which we have made. We have now more than 30 altogether. and we are very interested to see how powerful our technology really is.